So let's look at the central dogma, the idea that DNA is used to make RNA and eventually a protein. Now, we need to speak flowchart here. It's just when we get a lot of complexity, then a lot of times we break this down into simple terms, but it would certainly be easy to confuse what's going on here. It kind of looks like DNA goes through the process of transcription and becomes RNA. Well, that doesn't really make sense. You don't take a strand of DNA and simply turn it into RNA. No, you have the DNA, and then you use it to make the RNA, but in the end you still have the DNA. So it's not transformed, it just is used to make this as a code. And the same thing for translation. Okay, so that's one point of clarification. So we need to expand on that, and that's one of our main goals here. The other one is I want to point out something I have before, and that all these guys are nouns, right? They're things, they're physical objects that I just showed you, whereas transcription and translation are both verbs, they're processes, they're things that happen. And kind of like in a good news story, there's several questions we should ask. What happened? How did it happen? Who was there? Where did it happen? And when did it happen? So in transcription, remember that unlike replication, where we used the DNA polymerase, and its job was just to start at the beginning and copy the entire chromosome, there's a lot of DNA, and that process takes a while. But we only need to use RNA polymerase, and we're not going to start at the beginning. We need to find the right gene, and there's so much DNA, and we just need to find the right part. So we need a little flag telling us, hey, RNA polymerase, it's right in here. And so we might need to uncoil that DNA a little bit. This is so neatly packed, I'm not going to do that. But looking in at our DNA here, this is the promoter. So that's essentially like that little flag in there saying RNA polymerase, land here. And now we have two strands of DNA, but we only need one of them because RNA is single-stranded. So RNA polymerase actually does its own splitting, and in here it's just going to bubble out this little piece of it, not split the entire strand. And so we'll lay down our template strand again and start laying down complementary bases. So here we're going to we'll start right after the promoter, but we use uracil instead of thymine. And once we reach the termination site, then we stop, this goes away, that leaves the nucleus, and DNA remains safe and sound where it belongs, in the nucleus, nice and protected. Okay, so we focus a lot on these. That's the actual process of transcription itself. I'm going to expand that here in a moment. We also want to ask who was there. In other words, what enzymes, and we know that this is one of them. Where? Well, that's an interesting question. It involves DNA. DNA is found in the nucleus, so it's in the nucleus. When? That's a surprisingly difficult question to answer. Let's go back to a different process that we looked at before, and that is DNA replication, where we use DNA to make more DNA. Well, that happened in the nucleus, we know that. When? During the S phase of interphase. To be a little more specific perhaps, we could say it happens before meiosis or mitosis. Okay, well, when would you want to do this? When would you want to make a protein? And transcription is going to happen before translation, has to happen in that order. But when? Like, is there a certain time of day, a certain time of life? This happens basically 24 hours a day, 7 days a week, 365 days a year, from the day you're born until the day that you die. You always make proteins. And some cells are a little more active than others. Like glands that make enzymes are going to be cranking out proteins all the time. Skin that makes keratin and hair are going to be cranking out lots of protein. Just like replication is going to be active in your skin cells, where you do lots of mitosis to replace those worn out skin cells, but not really as active in brain cells because they don't replicate, they don't multiply. But both of them are going to be making lots of protein all the time. So the when is kind of an interesting question on this one. It's happening all the time. Various places in the body and in the nucleus, for transcription at least. Let's talk about the what and the who a little bit more. So when it comes to transcription, it's a little deceptive to just say this is one process, right? It's not just one thing. Like I said, you don't start with DNA and end up with RNA, poof, there you go. And so let's expand that out a little bit. Actually, let's expand it out a lot and say that we can break it down into three main phases. Now, that's not steps. And first is initiation. And for that, we're going to need our DNA promoter, right? So we need to know where to start. And so in terms of what's happening here, answering the who and the what and the how. So for initialization, we need a promoter. That's one of the ones. And that's going to help RNA polymerase find the right spot. And then we probably need some different nucleotides to begin that. So we got at least three players here during initialization, and a few steps where the promoter acts, RNA polymerase comes in, splits the DNA strand apart, and starts adding these. Now, the next phase, really the longest phase, is elongation. Obviously, RNA polymerase is still involved here. The promoter is not, but we need our coding strand of DNA, and we're going to need not one, but many of these RNA nucleotides. And so each time RNA polymerase matches one up using complementary base pairs, all right, we matched up an A and a U, and a G and a C, and a C and a G, and a T and an A. And so this is going to go on for quite a while, maybe several hundred individual steps. And again, remember, this is happening in the nucleus. So inside of a cell, you have the nucleus, and that tells you must have lots of A's and U's and G's and C's all inside of there along with the DNA so that RNA polymerase can do its job. So definitely the longest step. And then finally, we have termination. And so who's there? Should I orient this way? Here we have our termination site, RNA polymerase. We don't need any nucleotides for this one. When RNA polymerase reaches that termination site, basically it falls apart, as does the new RNA strand. And so that RNA strand falls apart, and that's what happens there. Right? So RNA polymerase is released from the DNA, and the new messenger RNA is released as well. And so that answers the what and the how this is happening, also who's involved, and where is this taking place. This is all in the nucleus at a specific gene. Now in eukaryotes, there's an extra step that's needed before this messenger RNA leaves the nucleus, One is on the 5' end here, the one with the phosphate. It gets a cap. Let's call that the 5' cap. I'm going to symbolize it that way. And then it needs a tail on the 3' end made out of a bunch of adenines. And so that's the poly A tail. Notice I've drawn a little tail in there. They don't really have them, but it just helps us remember. Now it can actually leave the nucleus, and remember, prokaryotes don't have a nucleus, so they don't need to do this. The what and the how are going to be very different. The who is going to be quite a bit different. Where is going to be different. When, pretty much the same. It's going to happen all the time, your whole life. So let's expand, and it's conveniently the same steps, right? But it's not just one step. We again have initiation. So it's going to be pretty important. Do we need DNA for this? Nope, we don't need any DNA. We're going to need that messenger RNA, and we're going to need that one fully processed with that 5' cap on there and the tail so that it can be recognized. And we need to get this in the right location at a ribosome. There, that can be a ribosome. Kind of hard to show a ribosome, it'd be much larger. And so we're trying to get the messenger RNA together with the ribosome, and we're always going to need to establish a reading frame. And so at the ribosome, we're going to find the complementary bases, so we're going to need our tRNA. Let me just move this ribosome off to the side to make this a bit easier. And we're always going to start at the AUG codon on the messenger RNA. You'll find that these are complementary and anti-parallel. Here's our tRNA, and see the little symbol there indicates that that one carries methionine. Your cell has its own process of figuring out how these guys go together and putting the right amino acid with the right tRNA. All right, so they come together, and now initiation is done. Okay, so let's sum up. Initiation, the ribosome parts came together, so did the messenger RNA, and the first tRNA with its amino acid. I don't know if those are all separate steps, but there's quite a bit to be done there. It's not just snap your fingers and there it is. And like before, the longest phase is going to be a lot of repetition and elongation, where we just keep doing this over and over. So who's going to be there? Well, of course our messenger RNA, and not only the ribosome and this tRNA, but that tRNA and its amino acid, and this tRNA and its amino acid, and they're all going to be involved carrying these amino acids into place. And let me move this RNA strand out of the way here. If they're both sitting here side by side and both these anticodons are matched up with the codon, then this amino acid is transferred to that one. And now we have a growing chain. This one gets transferred to that. And this keeps moving down the line in the ribosome. So that's essentially elongation. We add the next amino acid and the next one and the next one and the next one and so forth, each coming in carried by a tRNA. All right, so there's that phase of it. And then finally, we reach the termination phase. Fairly brief, but one of these is going to be what we call a stop codon. And so that's, instead of coding for an amino acid, it's going to say you need to stop. And so that brings in a very special tRNA with a release factor, not an amino acid, just a release factor. So who is here? This one, this tRNA, and this specific codon. There are only 3 of them. A lot of amino acids have 4 different codons. There are 3 of these that all say stop, and that causes the ribosome and the tRNA, the mRNA, and the amino acid sequence, the protein, to all fall apart. So that's termination. Basically, the right release factor comes in, everybody falls apart. So just really a couple of steps there, and we're done with that.